Hey guys, welcome to another episode of Let's Be Blunt with Montel. I'm of course your host, Montel Williams, where we explore the world of cannabis in its many, many facets. Now today, we're zooming in on the realm of cannabis testing and how innovative solutions are helping the industry evolve safely and efficiently. I'm so thrilled to welcome a guest today, Dr. Nathan Johnson, CEO of Verne Bio, with an impressive background in microbiology, molecular biology, and bioinformatics. Nathan has been a pioneer in developing cutting-edge testing solutions for cannabis and hemp. Nathan, thanks so much for joining us today on Let's Be Blunt with Montel, sir.
It's my pleasure. Pleasure to be here.
Absolutely. Let's go back, if you will. Let's start from the beginning. Let's start with your role at Verne Bio and how you found your way into cannabis and into this industry. What sparked your interest and led you to, creating Verne Bio?
Yeah. So my background is a little diverse. I actually started in cancer research as well as doing a food microbiology before going after my Ph.D. So I worked at places like Harvard Medical School, Dana-Farber Cancer Institute, other pharmaceutical companies. And my partner now, at the time, he did contract law for different cannabis farmers. And when the bill was passed in 2018, he highlighted just how much help they needed.
Farm bill. You're talking about the farm bill.
The farm bill back in 2018, because a bunch of the hemp farmers in Massachusetts got feminized seeds, but they weren't feminized. And so a lot of them were having trouble growing females when they were getting males and females. And so my partner highlighted, hey, all your background in cancer research, all those skills would be really useful in the cannabis industry. And after meeting a couple of farmers that were using cannabis to treat their cancer ailments, I was quite curious about how is that possible, because we didn't get taught that in school or in the pharmaceutical companies I worked in. So quite curious. And we developed Vern, where we focus on plant health using those different tools.
Wow, it's excellent. And I'm sure you started researching all of the research that have been going on for the last 50 years
Absolutely.
around the world and other places, which was really kind of leading the trend for us. And a lot of that research was funded by the United States. First of all, let's break this down a little bit, because being a cancer doctor, have you been able to discern, because there's lots of published, peer-reviewed studies out there these days, have you been able to discern some of the fact from the fiction about how efficacious some forms of cannabis have been and could possibly be when it comes to treating cancer?
When it comes to science, using like the scientific method, is you always start off with observations. So before doing an experiment, before doing any kind of research, you use your eyes, you observe what's going on. And what I've observed is I've seen patients that have cancer, they have visible tumors on their skin, and it goes away after a cannabis application. Why is that? Why is that possible? How is that possible? There are many, many anecdotes of different people doing this. So why is that done? Especially when you have reports out there that will talk about smoking cannabis is a similar carcinogen as smoking tobacco. And so there's a lot of mixed information on what exactly is going on.
Yeah, you said, that's what drives me crazy. I'm so glad I have a PhD on to be able to talk about it, because we can go all the way back to, the U.S. government's own patent application for CBD and other minor cannabinoids back in 1999 that they gave themselves in 2002, which is patent number 6603 507 or 6630 which in its abstract, it discusses how certain cannabinoids have affected, cancer cells. This was back in 2002. And you look at some of the research that was coming out of Israel back in the late 90s, there is research after research after research that is peer-reviewed, published documents stating the efficaciousness of some forms of cannabis or some of the cannabinoids that have had profound effect on cancer cells, especially some of them that actually thwart the, the development of certain, access to blood. Talk a little bit about that, because and you said you weren't taught this, but this is something that's been around, my friend, since the late 70s to early 80s. And we're now even still trying to fight the teaching of this to people like yourself.
Yeah, so let's do a little step back in terms of cancer as well
Sure.
as the different applications of cannabis. So
Sure.
in pharmaceuticals, how they approach most diseases is that they focus on one target, and that's just because the human body is extraordinarily complicated. And the proof that if you focus on one thing to get rid of it is a lot simpler problem than if you're talking like 200 different things at once, which is my prediction on how cannabis and cancer interact. And so, take the example of THC and CBD. There's definitely immune responses that happen with CBD. There are binding factors for THC, CBD to CB1 and CB2, which exist on almost every single cell in your body, including cancer cells. And so when you see that kind of uniform expression within your body, that indicates, as many people have alluded to, on the endocannabinoid system, or just the ability for your cells to respond to these different factors. Now, there is lots of research, as you said, and from Israel, United States, even some in Europe, that highlight if you have cancer cells in a dish, you apply different THC or CBD combinations, those cancer cells will dwindle, reduce, possibly die off. Now, the challenge with that is how do you take that observation, how do you take the observation that I've seen from patients and translate that into a mechanism of action, or how does it work? Because if you don't know how it works, it becomes very difficult to understand what is the limitations of it. So, for example, when I was at Dana-Farber, we did breast cancer. I did a focus on inflammatory breast cancer, and it was one of the most aggressive breast cancer types. And there was a lot of information on what did and didn't work, very similar to how cannabis and cancer. And the challenge with this is that when you have something that is so promising like cannabis, what happens if you do that clinical trial and half the people didn't respond? The FDA is going to say this doesn't work, but a pharmaceutical company wants to bias or wants to make that clinical trial exist with most people responding to that treatment. And so, in terms of the patent on CBD from the government, on patents in general, the reality is, I think there's a lot more to it than just simply THC CBD,
Well,
because if that
I mean, because we do
then you would
know
see the responses immediately.
yeah, I mean, and also, I'm sorry I didn't cut you off, but we
Oh
do
yeah,
know that there are well close to, depending on who you listen to, somewhere around 250 different cannabinoids. And when we start looking at the acid versions of all of them, and every single day or every couple of days, they come up with a new one from CBC
yep,
to CBD, CBDA, CBD. I mean, it's a continuum, something that should have been researched for now 50 years, finally starting to slowly be researched. And I would probably agree with you 100%. I think, I'm kind of a believer in science in this respect that, for every action, there's an equal and opposite reaction. And so I think for every disease on this planet, there is somewhere an existing cure or somewhere an existing, methodology to reduce the effects of that disease. And, some of them we've destroyed by burning down half the rainforests in the world, but we still have plants like the cannabis sativa plant, the hemp plant, that has so many active ingredients and components. I'm just excited about the fact that there is AI now where we can speed up the process of research by, you know, reducing volumes of information to come up with some sort of solution. So, I mean, I agree with you that I think we're just scratching the surface when it comes to researching this plant.
100%. Yeah, and that's one reason why I left my normal job in cancer research and joined the cannabis industry, because as I saw it, there's not very many opportunities in a career for you to join a new emerging market like this. I kind of think of the cannabis industry similar to other biological research fields like microbiology or other scientists back in the 50s and 40s when different things came about for the first time. There's a lot of innovation, a lot of things that need to be understood, and it's going to take some time to figure out the nuances of this. guarantee in 10 years from now, the conversation is going to be quite different on what is and not known about the plant. If you look at the research body for cannabis, it is exponential in terms of the papers published around this plant, and it's going to increase on that because there's a lot to know. That's one reason why I think companies like Pfizer, who have bought patents on doing these different synthetic forms, are so powerful because it allows them to look for different small molecules that may have an effect. And then the additional research on cannabis will allow that research to continue on understanding that nuances as well as the combination of the different molecules.
So tell us a little bit more about Verne Bio's mission and the unique capabilities and solutions that you're going to offer.
Yeah, so our mission is to help the smallest to largest farmer in cannabis in terms of their risk, de-risking their operation, as well as increasing their yield. Plants, just like people, get sick, and a sick plant is not going to produce as well as a non-sick plant. And on top of that, because organizations such as the USDA or decades of research doesn't exist for cannabis, we're largely blind on what's going to infect cannabis. For example, hopsylate viride, which was identified back in 2019, it came from hops. It jumped from hops to cannabis, and it causes about $4 billion of damage in the cannabis industry on a yearly basis, is the estimate. That is just one of possibly hundreds of pathogens that are going to be found in cannabis that cause different harm. And what's the cannabis cultivator going to do without having the tools to both detect as well as to get rid of these different pathogens? And so that's what we're helping with. We come alongside these cultivators, identify novel infections, as well as solutions on how to detect and get rid of it.
It's
And
very interesting. As I am, where are you in every state? We have 38 states in the District of Columbia right now that have some sort of cannabis law that allows for consumers to access cannabis. How many states are you in?
so we have a lab in Massachusetts. We have boots on the ground in 6 states at the moment, and we are expanding beyond that.
Nathan, I'm been doing this business now for like almost 24 years, and I've been kind of taken aback by the fact that, in a lot of states, I'll get a product tested, and then a week later, maybe for some reason, the individual contract manufacturer I'm working with has a different lab tested, and it comes back with different numbers. When is there going to be some consistency in the lab testing world when it comes to cannabis?
So you have three different problems there. One, the testing standard is not the same. So lab A versus lab B, how they actually test may fundamentally be different, and it can be wildly different in terms of results. And that's largely due to the lack of standardized testing protocols. It's not that there's not approved testing protocols. It's just that when you have 10, 20 different protocols approved, and you can pick and choose which one you want to use, you can get different results. Just the fundamental nature of that. The other challenge is the consistency of the plant. So every single trichome is a dynamic amount of cannabinoids it's making. And when you have bud on the top of the plant versus bud on the bottom of the plant versus plant A versus plant B, and so on and so forth, there's a high variability in terms of what bud is making. So the other secondary challenge is what is the real answer is kind of highly subjective. And normally, when you have that kind of variability, you're going to create a homogeneous or a very pure mixture that's going to be consistent no matter where you sample it. But because no one wants you to take all the flour and dice it up and make it one big bag of flour mixed up everywhere, that would be the only other way to make it more consistent and uniform throughout the entire protocol. And then the third area is the humidity factor. That's a huge factor with these plants in curing. While there are some labs that normalize for that, there are huge variation because of just how much moisture is in the plant.
So now, are we going to get to a point where we're going to have to try to figure out? You guys are going to end up having some sort of competition among all the labs to figure out who comes out on top, so that at least only you keep testing at the same place rather than multiple places. What's the solution to this?
So when I worked in pharmaceutical, you had the same problem when you did the clinical trials. So in clinical trials, you have to have usually a test to identify what patients belong, as well as what's the indicator on whether or not the treatment works. And what's interesting is usually those companies, they don't have 10 different labs giving them results. They just have one lab giving them a result. And the reason is because that number that comes out on the other end fundamentally doesn't matter. Why does it matter if it's 22% versus 24%? What the person, the user wants to know is what value correlates to what they feel or what they want. If it's 5, 10, 20, whatever, as long as they can get a consistent value that correlates with what they're looking for, that's ultimately what you're looking for. So like alcohol percent. So how much alcohol you have in your blood has a strong correlation to the cognitive ability of the driver, for example. THC, you don't have that, unfortunately. And so how much you have in your blood doesn't necessarily correlate to how well you do, because there's a lot of other factors beyond just that percent that changes what you can interact with. And so it makes it very difficult to come up with a universal rule on what test result you should do, because also the form factor makes it different. Are you testing chocolate, a beverage, flour, oil? All those create different nuances on the test result. And because there are so many different ways that you can do testing, until we come down to this is how you test for, say, E. coli. We have a very standard process on how you test for E. coli that was established in the food sector. Food microbiologists, we had one testing protocol on how to do this. There's different ways to test for it, but the fundamental fact is there's a pretty universal way on how to look for it. And so until we can get to that point where each state doesn't make up their own rules, there's more of a universal rule set, you're going to constantly see this problem, as well as giving the labs the flexibility to choose whatever they want. That's going to be the other challenge as well.
And it's very interesting because these new testing solutions that you've been coming up with are really going to try to transform the entire industry. However, if other people don't use your facility, then they won't get the same transformative effect, right?
That's right. Yep. And we have that exact problem with some of the pathogens we test for. So our test for, say, Hopsite and Viroid is one of the best in the market. There are other tests out there that are not as good. So, for example, if you use that test result as a decision, so say you have a plant that's tested for the viroid, and one lab says negative, it's not detected, and the lab says it is there. That's because there is a real reality that the assay is not as sensitive in one group versus another. And so it makes it very challenging to deal with a situation when you get incomplete results. And that is just the nature, unfortunately, of science and testing.
And this is what you have. You got these things called eye test kits. Is that what you're kind of talking about?
Yeah. So the eye test and we test in our case, it gives you the ability as a grower to do the test yourself. But there's always a limitation on what you have access to. For example, the equipment you have access to isn't the same equipment that we have in the lab. And so the testing ability that you will have will never be as good as a lab because of that limitation. Now, on the other hand, you get a quick answer.
It gives you kind of a heads up.
Yep. So, and that's why we made both those product lines, is the ability for, if you see something weird, you think it's this, you want confirmation, is it this or not? But if you're looking for something that's more comprehensive and more sensitive, then a lab's always going to be the better alternative.
Well, what are some of the specific challenges that you encountered when you were developing this eye test kit, and how have you overcome them?
So the first challenge is there's no golden standard on what we're testing for. So if I work in another industry, if I work in, say, cancer research, there are actually companies that will store cancer cell lines in vials in liquid nitrogen. So I can literally just call them up and say, hey, I want to test breast cancer. Can I have a cell line that does that? Call them up, pay them a couple $100, and I have it in my hands in a couple days. Same thing with mice, same thing with rats, same with all this other infrastructure that's normal for researchers. When we joined the cannabis industry, there is no one we could call up for, say, Hopsite and Viroid in terms of that infection. Now, there are plenty of growers who are doing it unknowingly, but how do you evaluate a test if you don't have any way to know what's real versus what's not? And so one of the first challenges is just trying to build your own infrastructure as a company on things that you got used to in other industries. That was one of the big bottlenecks when we first started.
And that's one of the things that Verne Bio is kind of doing. You're starting to like leverage some artificial intelligence to improve your testing accuracy and efficiency, right?
That's right. Yeah. So We are trying to get to the point that as a cultivator, when you are working with your crops, fundamentally you do not know what's really going on. You have some guesses based on media or what you read, but in reality, you really don't know what's there. And so we're as a company, we're trying to maximize how many things we can detect at once, without having to increase the cost. And so the challenge with that is it's actually a factorial math equation. What I mean by that is when we have 16 different targets, it's 16 to the power of 16 in terms of possible combinations. And every time you add another target, it adds that number on there. So if you go to 32, which is our ultimate goal, 32 to the possibility of 32 power is how many combinations are, which are there's millions upon millions. And you can't run that many experiments. You would go broke trying to run all those experiments. And so you need a way to identify what's a good experiment versus not. And so we use AI to look at all those combinations to identify here's the top 10 experiments that should work. Run those rather than running all those other millions that are out there.
And that's some of the things that we can expect from Verne Bio in the near future,
That's right?
You're developing
Exactly.
this up.
Yep, we're trying to increase as many things as possible because we know there's hundreds of pathogens that are infecting cannabis, and we know this is causing harm, and growers are blind to it. So,
and we also do know that, again, I talk to people sometimes and say, well, I really like this strain. Well, I say, where'd you get the strain? And they'll say, well, I went to the store and I got another, I like it because I can go to another place and it's consistent. I say, no, it's not. Just because you bought something that was under some name in XYZ dispensary here doesn't mean that it's the same product in another dispensary, because we do know that, even down to each individual plant. I mean, every seed, it depends on the growth cycle and the grower and whether or not they were watering at the right time, whether or not they were adding nutrients, what kind of nutrients are added. I mean, anything can affect this plant, correct?
that's correct. And there's actually competitions around the country that actually leverage this. They will give the same seeds or the same clones to, say, 100 growers, and they'll have a competition who can grow the best plant. And they'll literally have 100 different results based on how you grow the plant. And so when you have that diverse of an organism on the environment effect, so there's always the idea of nature versus nurture. How much did nature or how much did your parents' DNA give you versus how did they bring you up? Same thing with cannabis.
But I mean, should we not be able to, even if those 100 different clones that were used in 100 different places, if I got a valid COA on all 100 of them from the same company, I, as a consumer, should be able to read the COA and find the one that's closest to the one that I achieved the results I was looking for with, let's say, a different plant. If I find that again, I should probably achieve the same results. Maybe, maybe not, right? Because that'll also depend on what I ate,
Who you are depends on what that COA contains, because they're not testing for all 200 different metabolites that could cause an effect. They're only testing for 10 to 20. And so while there is some correlation, some data analysis that you can do to correlate that data to your effect, it's not the full answer. There's also no information on how you process things. Like, for example, do you have a mutation in your ability to synthesize THC or metabolize THC? So how you process it versus how I process it is going to be different. If I smoked yesterday versus today, I'm also going to have to react to it too differently.
Because that's also very important. I really like the fact that you even bring that up, because I have a product that I put in the marketplace in the Massachusetts area, and I for a couple months I felt like this particular batch seemed to be consistent in what I derive from it on a daily basis until, let's say, day 14, 15. Then I started realizing, wait a minute, it's not the same as yesterday. I thought it was, but then tomorrow it's almost the same as it was yesterday. Then I look back at yesterday and realize that, yesterday I did have a gummy, and I also hit my vape pen. So that changes everything about my own biology when it comes to how my endocannabinoid system is going to absorb what it is I put in, right?
That's absolutely right. And also, the more you consume THC, the number of receptors in your cells also decrease. So if you, and that's why there's THC tolerance for a lot of people who use it every day, and why the whole concept of not using it for a couple days or a week or so will change your whole experience altogether.
Oh,
And
no question. Yeah, that's a fact. Yeah, yeah. But I mean, this is again part of this whole process that I've talked about for years now, and I'm even working in a couple different marketplaces around the country where I'm trying to explain to people that, we need to spend more time. I mean, first off, well, your business is a B2B business, period. I mean, you do B2B. But we need to translate some of what you say from a B2B standpoint to a B2C standpoint so the consumer can understand a little bit more about why, I had that batch and I've had it in my drawer rather than my refrigerator. I had it over here, you know what I mean? And I stuck it in the bathroom or, whatever you put it at, which will change the composition. And as the plant actually decays over time, its composition changes also, right?
and you have just explained why we started with Vern and not gone after the consumer experience. Because I, as an individual, as well as my team, I want to learn better on all those nuances of the plant. So that way, when we try to go after how does this flower react to this person, we have a better context as to what's the possibilities. Because there is so much complexity on who you are as an individual, as well as how you've interacted with the plant, versus what the plant produces, versus what the plant variability on what it can produce, as well as there's nothing to tie it all together in terms of this plant is actually this strain. Because as you alluded to, companies are going to name their different strains, different whatever they want to name it. Honestly, sometimes there's some correlation to it. Sometimes, like it's from a Kush line because it has that word or a paraphrase on it, and sometimes you don't. And so, how do you know as a user what could possibly work with you?
Yeah, yeah. I have been really, literally, I think I tick a lot of people off in the industry because I keep trying to say, stop with the silly ass name. Excuse my mouth, but stop it because you give people the impression that if I have, I don't know, applesauce in one market, it's going to be the same plant in another market, and it's not. It's all, it's just like, when you talk about how come you like this pizza better than that pizza, because it's the hands. You know what I mean? When you go to a restaurant, why does one bowl of lasagna tastes 10 times better than the other bowl of lasagna? It's the people's hands that actually made it. Maybe that one guy, shook the shaker 3 times rather than 2 times. It's so subtle and so nuanced that, even when it comes down to, we see it all the time now. I go into dispensaries and I'll hear one of the tenders talking to a person and they'll say, well, you probably would like to have more of an indica experience than a sativa experience. And I want to reach across the counter and say, stop saying that because half the plants that we have right now are hybrids of some sort. They have been crossbred, that. I don't even know if those terms even mean the same. Yes, we will find some short, bushy plants, and yes, we'll find some taller plants. But the taller plants have bushier buds, and it's like because they were crossed and crossed and crossed and crossed. So we should throw those terms out and start looking at it from, I think, a purely COA standpoint, where I can take a look and see on a COA that lists the terpenes, understand that that's the variable that I didn't look at when I bought it over here. But let me look at it again because now all of a sudden I got an OG something that that's not the same OG something over here. If they use the same laboratory, clearly these two COAs are different. So it's almost like we need to start educating the consumer more. I mean, first of all, I think that's what's going to be imperative to keeping this industry robust is that we have to start reaching out to the consumer and letting them try to understand. I'm not trying to make you a scientist, but what I'm trying to make you understand is that certain things may affect you differently than they affect other people. And if you look at these numbers and you find consistency in those numbers that give you kind of an overall consistent response, if that's the one that you were looking for to help you feel better, whether that be medically or, we call it adult use wise, then that's kind of where you probably want to land. I don't know. Am I making it too complicated for a consumer?
No, you are actually alluding to one of the projects we tried a number of years ago, where we try to take the different COAs and try to bin it in different buckets or different categories on what could actually work with a consumer. And the challenge with that is you have all these cannabinoids and terpenes, whether you use like say the top three terpenes in terms of the outcome, or the other fact that what you smell may not be all only terpene. There may be another compounds that are in there, and as well as the cannabinoids. They're only quantifying a few of them, and so the what we have found, or when we were working with different patients, is you can get pretty far with that data. It's a lot better than just guessing. That's for sure.
I
But in
go
terms of consistency,
when
yeah,
I produce products that I have gone after, taking distillates, putting them back together again, and even if I'm using let's say a rosin and recognizing that's the whole plant, but if I add a little bit more of, let's say, a little bit more of linalool, a little bit more of beta caryophyllene, that seems to tweak the feeling that I have a little bit differently than had I not put that extra in there.
absolutely.
Yes, good.
No, it says it absolutely. And what you're describing is the field of pharmakinetics in terms of how does all those things interact with your body. There's actually a whole you can get a whole degree in that in terms of that complexity. And so it's first off, until you can get a lab that's consistently going to give you that COA, that's the first step. The
Right,
second step is how do you understand the nuances, all the different cannabinoids and terpenes? That's also the other complexity. So how do you know if it matters if you have this amount THCP or THC or whatever versus this CBD or this CBD variation
right.
plus this terpene? And so on one hand, people are going to become like wine connoisseurs when it comes to
Yeah.
cannabis, for sure. But it's also way more complicated than wine connoisseurs, in my opinion.
I almost think I agree with you 100%. It's almost like you become a sommelier because you have to really kind of, it's like with me, I formulate products based. Um, I've got a couple of products in the marketplace where I believe very strongly in adding CBD on the top of, let's say, my cart. I have a particular vape cart that's like 9 to 1 THC to CBD by volume, and then I put particular terpenes on top of that to see if I can help reduce that anxious effect that people get from a high THC product. Well, consistently, I found that the people who have used my product come back to me and say the same things over and over again. I didn't have the jitters. Though I got as high as I could possibly be, I didn't have the jitters. I wasn't so anxious. And I've noticed that with my product uh, consistently over time. It's like sometimes when I'm out in public, if I take something that's too high in a THC, it really gives me kind of like the jitters and an annoyance. Like I'm looking around behind me and all that. But when I take my own product, I don't have that edge. I'm literally, I like one of them in particular to go to the movies because I can sit in the movie theater and just, you know, be completely blasted by all the imagery and all the noise and all that. But I'm not afraid if somebody walked down to go to the bathroom. You know what I mean?
Mm-hmm. Yep. And that is exactly what you're describing in terms of the entourage effect that can have that different major effects. Small changes can have major effects.
I believe. Now, can you discuss a little bit about the importance of sustainability in your testing solutions in your practice?
So, in terms of sustainability, the first one is if you have plants that are not healthy or they're sick, you're going to throw away those plants or all the wasted energy as well as the substrates that you use. So, for example, a lot of people use Rockwell or cocoa. Um, Cocoa is from coconut hair. Rockwell is from basically volcanic rock. It's not really reusable. You have to throw it away. If you visit these large cannabis cultivation facilities, they'll have dumpsters of trash that they have to do. Some states, they have to let the Cannabis Control Commission review all that. And so just the very fact of if you don't get your plants to market, you've wasted all that energy, all that resources. On top of that, while our testing obviously has some plastic usage that has to get one use alone, there is more and more substrates that are starting to come to the market that are economically advantageous and can replace these different substrates. And so we partner with those companies because we want to have sustainability. We want to improve that efficiency, especially indoor cultivations. They use a lot of electricity and a lot of water. And ironic considering that this industry got started off people who were trying to be very green, and a lot of them are not that way. But I think as the industry goes forward, like I saw a facility in Massachusetts that they have a completely 100% water retention in their facility. They recycle everything. Now that creates problems because any pathogens or any infections are in that facility now, but also they have very minimal water loss. And so it's a pro and con in terms of that usage, as well as people that use solar panels for their cultivation facilities to reduce electronic usage or electricity usage. So there are lots of groups that are trying to do all these things. But by and large, most facilities are not well designed. There are a lot of repurposed buildings that have a lot of efficiencies in them. And so, but in terms of sustainability of our testing solution, as well as what we do, by having healthy plants that get to market, that achieves a lot of lack of throwaway from there alone. We had one facility that threw away 300 pounds because of just how much infection there was, for example.
Sure. Well, can you share some of the success stories you've gotten from farmers who have been using your solutions and how you've helped them?
Absolutely.
Sure.
We have had many cultivators that will usually call us when their plants are fallen down or they're sick. And it usually takes 6 months to a year to get them righted again. But we've had many success stories of people have high degrees of infections that are now less than 1%. It's usually hard to get down to 0% because that pathogen is usually around the environment somewhere. But the ideal goal is to get to that level of very, very low infection and also monitor how the facility is running. We have facilities in California, Massachusetts, Rhode Island, for example, that have used us to help right the ship in terms of that infection. And unfortunately, what usually happens is that it's from a breeder or from an outsourced plant material that they brought in that causes the whole problem to start over again.
Wow. And I mean, I guess, for the consumer, we don't get to know what
unfortunately
the grower's gone through to get product onto a shelf. And then, we end up consuming some of the stuff that either doesn't have a good COA on it or, doesn't let you know about some of the contaminants that this place has gone through.
Yeah. Yeah, there's actually a few companies that are starting to have remediation certification to give to different companies that say no to remediation. They don't have to remediate or use x-rays or whatever on their product to get to market. And that's actually one thing that's slowly changing in this industry. When we first started back in 2021, we really couldn't tell anybody who we worked with. We weren't allowed to because they were so afraid to disclose that they have infections and that they hired people to help with that. But it's starting to shift. Even now this year alone, we're starting to take pictures of the facilities that we go into to highlight, look, they're trying to do right. They're trying to fix this. They're trying to make it so that everything's healthy and beautiful.
When you walked out the door, everything was.
You walk out the door, you say, look at how much care they do to take care of your plants. Because unless you go into these facilities, how would you ever know? How would you ever know that you put on a full bodysuit to take care of the plant?
That's something that I think you guys ought to put out your own Verne, publication, telling people, when you go in the stores, here are the people that we've worked with to help remediate their problems. And we know that when we walked out on January 1st or 2nd or February 5th, these guys were actually producing a product that, we willing to say was was good.
Yep, exactly. And that's, While there's still a lot of companies that are hesitant about that because obviously they don't want the wrong messaging, but there are plenty of people as time goes on that are starting to say, yes, it's okay to talk about that we have this problem and we fixed it.
It'd be okay to talk about getting a Verne seal of approval too.
That's exactly one of the other products that we're talking about with different cultivators is to have like a Verne certification program. Because largely the plants that they bring into the facility, because they're constantly trying to rotate the number of strains that they have, create a huge risk. How do they know that those plants that they got from another breeder will actually perform the way they're supposed to? How do you know they didn't come in with an infection?
How is the Fed? Has anybody in the Fed reached out to you guys to see if they could bring you aboard to help them navigate what they're about to navigate by, I don't know. I would ask you your feelings about, the rescheduling process, but I mean, no matter what the Fed does in the next 90 days, they're going to have to either reschedule or, I know that hasn't been held up. It got held up for a little while because, there's this group of legislators who signed some petition to stop the DEA from rescheduling. But I don't believe that's going to be able to stay in place for a long period of time. So they're going to have to do something. But when they do so, and let's say they reschedule to Schedule 3, that's going to bring this under the purview of the FDA. Has the FDA or anybody like that reached out to you to say, we want you to be a part of this transition?
No, unfortunately, no. But I also would highlight that we've never had a Schedule 1 drug like this to have such differences between different states. And I would say that descheduling to Class 3, how would we expect that to be any more or less complicated than it already is? And
Well, I think it's going to be ridiculously
it's going
complicated.
to be my advocacy is that any medical patients or any medical cannabis has to have different standards than recreational. Because presumably that medical patient could be immunocompromised or various other factors. And so their cannabis should be much cleaner than, say, recreational. So that's my prediction on probably the most fundamental difference that's going to happen out of all this. But in terms of all the extra stuff, your guess is as good as mine in terms of what they eventually settle on. And I think it's going to take years. So
I agree with you. I think anybody who thinks that all of a sudden, this is going to be like, hey, everything's perfectly fine now. They're crazy. They're crazy because, they just have to stop and remember Schedule 3 drugs right now by law needs to be dispensed through pharmacies, not dispensaries. So they're going to have to clear that whole thing up. And you got any final thoughts, my friend?
Final thoughts regarding descheduling or in the whole conversation?
How about the scheduling and then the conversation about future? What's going to be the future for your industry? Because I think, you guys are setting a standard that anybody out there faking testing should be trying to follow. I hope you'll get in all 38 states very soon. Are you in Georgia yet?
I'm not in Georgia yet, but we definitely want to get Georgia.
Huh?
Definitely want to.
Yeah, well, we're working in Georgia right now with one of the two big licensees, and so I'll make sure I recommend your services to them.
Well, thank you very much.
Sure.
Yeah, I'm right there with you with descheduling. It's going to be super complicated, super many discussions. Lawyers are going to be the winners for now as I go through all the hours to figure out, like you mentioned, pharmaceutical pharmacies versus dispensaries and state by state in terms of different rules and legislation. So it's going to be super complicated. But in terms of the standard going forward, absolutely, there is a lot of information to dissect with cannabis. There's a lot of nuances as to understand what should be the thresholds for different things. For example, different states have different amounts of lead that's allowed in cannabis. Now, there's not different amounts of lead that the human body tolerates, but there's going to be some nuances there as to what they allow. And so all those different things are going to be figured out in the upcoming years and need plenty of people to figure it out all together. It's not going to just be just one lab. It's going to be all of us together.
Well, I got to tell you, I hope you take me up on this idea, my friend. I think you guys ought to be putting out a book so that, those who are consumers, again, can feel a little bit more comfortable about, at least even reading a COA. You know what I mean?
Yep, absolutely. We could do something like that for sure.
Cool. Well, I got to thank you so much, sir, for being a part of the show today. To all of my viewers out there, I know they're going to love this interview, and they're going to love the fact that there are people out there looking out for their well-being. So keep doing what you guys are doing, and for all of you out there, keep doing what you're doing. Stay educated, stay frosty, and stay really curious about what it is you're putting in your body. Okay. All right. Join us on the next Let's Be Blunt with Montel. Thanks for joining me on Let's Be Blunt with Montel. Please make sure you're subscribed and hit the bell to be notified when new episodes post each week. We'd love to hear your feedback also, so please send us your comments.